Okay. Hello. This is a review for POD 150 exam. It's not all of it, it's some of it. We have the nucleus. In the nucleus, we have a lot of things going on, but one of the main things that we talk about is the nuclear pore complex, or NPC. This is how proteins come and go from inside the nucleus. What do we talk about first? So there are different components. There's the core scaffold, which is just composed of rings. There are outer rings and one inner ring. There's an outer ring on the cytoplasm side, and one on the nucleoplasm side, and then an inner ring, and then they are anchored by, there we go, linker loops, which they link them together. That does not make them part of the core scaffold, but they do link the core scaffold. Linker loops also link FG loops. Those are kind of in the middle. So this is kind of like a flat view, and this is like a looking into the nuclear pore complex view, like if my eye was looking this way versus slicing it, and that's what we get. I guess it's not really sliced because it has the whole ring. This does not include the other components that we talk about here. So these are cytoplasmic filaments. We'll talk about those later. So the linker loops, I never wrote this on the board, but there's also like, if we have our full NPC here, there's also a membrane ring on the outside that like snatches its waste and integrates it in the membrane. So imagine that's drawn somewhere. Okay, so we have these FG loops, which make like a gel-like matrix and act as a filter. They are looking for one thing, and his name, its name, their name, her name. The name is important beta. We'll talk about important beta in a second. But the gel-like matrix makes it so that like some things can exit, some things. It's not just like you just go through. You know, you gotta crawl through there. So who crawls through is important beta. Important beta, she helps import. Yes. So say you have a protein, and you want her to go to the nucleus. She's a transcription factor. It's time to make something. To get to the nucleus, you have to be accepted by this person. To be accepted by this person, you gotta bind to important beta. First, you will be recognized. Your NLS, your nuclear localization signal, will be recognized by important alpha. And when important alpha binds to you, your nuclear localization signal, it'll free the important beta binding domain. And then important beta be binding. So now you have all three of these guys. You got your protein, you got your important alpha, you got your important beta. And I wrote ran GTP here, GDP, because it'll be ran GDP in the cytosol and ran GTP in the nucleus. And so when it's bound to ran GTP, which is a high ran GTP in the environment in the nucleus, that tells it it's in the wrong place. Because if I'm important beta, I gotta go collect more proteins to go back into the nucleus. So it'll exit when it's bound to run GTP. So now we have entered in, we are in the nucleus. Success. What are the other parts of the NPC that I can talk about? On the inside, we have the nuclear basket. It's one of two things that tethers to DNA. And so there's chromatin on the inside, and it can have effects on transcription. And what it does, it'll recruit chromatin remodelers. So here I have like histones being rolled away from this like site where ideally transcription would happen. Two, it would recruit transcription factors. So now that the DNA is accessible, we gotta transcribe it. And now we have our pre-mRNA, so we're gonna splice it, so it'll recruit all sorts of splicing machinery, and we'll get some mRNA. And then mRNA, if it wants to be exited, it'll go through nuclear pore complex, right? Anyway, then we have cytoplasmic film, or would it be able to diffuse, do you think? I don't know. Anyway, that's not the point. Final component is the cytoplasmic filaments. There's also 8 specifically of these, and also the basket pieces. I don't think that's something you need to memorize. Cytoplasmic filaments be grafted onto proteins. #RANGAP. That's all I have to say about that. Okay, we talked about one thing that tethers to DNA, but another thing is PNET2. So let's take a big look at the nuclear bilayer here, or double bilayer. The nuclear, the nucleus.
Membrane.
The membrane. Okay, so I'm gonna just skip to this thing. This thing is called PNET2. It's a chromatin tethering factor, and it'll tether to chromatin. And it can release it in certain environments, and that can change transcription because once it's released, it's available to be transcribed. You also have other things going on here. So if you have your nucleus in the cell, you, as a plant, can actually move your nucleus around, and that means that you have to be attached to something that's moving your nucleus. and that is the cytoskeleton. So you have these linkers of cytoskeleton and nucleus, probably linkers of nucleus and cytoskeleton link. And in plants, this is cache and sun. Cache is on the outer nuclear membrane, cache on the outside. Sun is on the inner, because it is, because it's not cache. And they interact, and the sun is attached to the nucleus skeleton, and the cache is attached to the cytoskeleton. So these are these rigid structures, so you can move it around. Because if it's just like not chained to the membrane at all, then it'll just kind of like, I don't know, I feel like it would just like rip away. But you got this rigid nucleus skeleton, which also defines the shape of the nucleus. So that's interesting. Also, kind of just like a thing to say, plants and animals do open mitosis, unlike yeast. So the nuclear envelope is degraded, and the DNA is free. Moving on. Endoplasmic reticulum. We have made some mRNA over here. Back when we were talking about nuclear basket, it's going to go get translated. And that will happen via a ribosome. So in most cases, you'll have a ribosome heading to, I don't know in most cases, in 2 of 3 types of things you can have happen. You'll head to the rough endoplasmic reticulum. If you have a transmembrane protein, that's hydrophobic, so you don't want to be secreting it or synthesizing it in the middle of your cytosol, because then it's going to like get all freaky and it's not going to like that. So it's going to be secreted directly into the ER membrane, and so it never has to be in a hydrophobic environment or hydrophilic environment. It becomes in the hydrophobic bilayer. But you can also have things that are hydrophilic be made inside of the ER, and those are the ones that are wanting to be secreted out into the plasma membrane, and they get put into the ER lumen. Also, if you have any questions, you can ask. Real people, no people. I don't know if I'm responding to any comments, guys. So these soluble proteins will be happily inside the ER. There's like, it's a matrix, you know. But if you are just like something in the cell that is just like a piece of a protein, maybe like a heat shock protein, I'm sure those aren't too hydrophobic. Yeah. You can just have that in a free ribosome. It doesn't need to be in an endoplasmic reticulum. So if I am a ribosome and I have a thing that needs to be gone to the ER, I'll have my whole sec pathway. We're not really going to go too deep into that. But basically, it'll be translated into the ER membrane through this translocon. Translocon will open a lateral gate for the protein to embed into the membrane, and then according to this diffusion retention model, it'll just like swim around until it gets to the place it needs to be. Obviously, this isn't the case if it's supposed to go someplace that isn't attached to the ER, but if your membrane protein is supposed to live in the ER or the nucleus, it'll just kind of like diffuse until it's tethered by something. In this case, tethered by the nucleus skeleton in my little diagram. And this is the ribosome leaving. The ER is composed of tubules, cisternae, and 3-way junctions. Tubules have, well, also cisternae apparently, but they have like their curvature induced by a reticulon, double hairpin structure situation. And 3-way junctions are made by RHD3. You can remember because 3-way junction. What does it do? It makes protein. It also stores calcium. I feel like this is more of a smooth ER thing, but we're just talking about ERs in general. In plants, they are pretty uniform. There's not really a huge distinction between a rough and smooth ER. And they store proteins. I'm just gonna move on from there. Also RHD3 that makes the 3-way junctions. It has like it is a transmembrane GTP ACE, and they will dimerize and do GTP hydrolysis, which results in diffusion. Moving on. Let's say you are an endoplasmic reticulum, you've been making proteins, and you notice that a lot of your proteins are unfolded, and that's building up. That sounds like a problem. You have to do something about it. Sounds like you're under some stress. Maybe you're in heat stress. You don't have enough chaperones. So you're going to initiate UPR, or unfolded protein response, in which this guy named Vic will detect an unfolded protein, and it'll say, that is bad. And it'll go to her friend, BZIP28, and it will say, hey bestie, we got a problem. And BZIP28 will aggregate. There'll be like a bunch like pushed together, and then it'll butt off in a COP2 vesicle, and it'll be sent to the Golgi. Quick stop at the ER Golgi intermediate compartment. We'll talk about her a little bit more, but not that much more later. And there, part of BIP28, or BZIP28, this part will be cleaved by S2P. And this part is a transcription factor. So that's going to go into the nucleus and increase the transcription, induce transcription of stress response genes because we're under stress. So like a chaperone, more chaperones, we can have unfiltered protein response, respond to them, make more chaperone. But let's say we have so many and they're starting to like aggregate and they're starting to like damage other cellular materials. The hydrophobic regions are exposed, things are going crazy. It's like, hang on, we don't have time to just make more chaperone. Like we gotta like kill this because if it gets too aggregated, we can't even destroy the proteasome. So this is our emergency mode, our ER-associated degradation pathway, in which case the bad protein is recognized by a transmembrane E3 ligase, ubiquitinated because that's what E3 ligases do. And that means it'll be able to go through this ubiquitin-specific ATPase CDC48 and send to the 26S proteasome. No more bad protein. Cool. Back to, so if you have a cytoplasm.
Sorry, do you have CDC48 being a transmembrane protein?
Yes.
No.
It's,
Pretty sure no.
how does it get out?
Slight change to what we had just discussed.
Yes. Okay, so this E3 ligase is going to ubiquitinate the bad protein. I'm going to draw ubiquitin coming in from over here, and it's going to send it through CDC48, and then it'll be delivered to the 26S proteasome. CDC48 is an ATPase, it's going to like pull it out of the ER.
And CDC48 is not a transmembrane protein.
Yeah. Thank you, Allison.
Yeah, okay.
Okay, so if we have back in time, we can make secreted proteins. We're going to secrete them. So how do we do that? We need to have other things happen to us. So we've been made, we are a protein, but we are not ready to be out into the world yet. We need to be translation, we need to have post-translational modifications. So we'll be releasing a COP2 vesicle. And you're like, what is a COP2 vesicle? How is that made? You have this enzyme called SAR1. Guanine exchange factor, B exchanging guanine. We are now active, SAR1 GTP. It's going to recruit the inner coat of a COP2 vesicle. And that's made of sec 23 and 24. And then it'll recruit the outer coat, sec 13 and 31. Now we have a COP2 vesicle. It's like, yeah, and at some point it'll be, and then it can dissociate and you just have this thing, which is still called a COP2 vesicle even though COP2 is not present. So now we are ready to go to the ERGIC, the ER-Golgi intermediate complex. And then we go to Golgi. That's all I have to say about that. Where glycosylation happens, so if you need to be glycosylated, the time is now. This is anterograde transport. You'll go from the cis to trans Golgi, which is more mature as you go along. Different layers can have different enzymes, and then you have some options. You can, if you need to go back to the ER, you can cop one vesicle, get your way back then. Also, both of these move via actin, unlike microtubules, which is what animals typically have, and actually it's a little bit faster. So actually a little bit faster. Anyway, moving on. Where to go next? Lots of options. So you'll next go to the trans-Golgi network / early endosome, which is also a sorting station. So if you have things coming from outside the cell, it will also sort things here. It can say, I want to destroy it, and then you go eventually to a lytic vacuole, or I can keep you and it'll just go to a recycling endosome, and then go back to the plasma membrane if you got from here. We'll talk about more about this destruction pathway in a quick second, but let's just quickly go over how you are able to get something from the plasma membrane. This is our retrograde transport. So endocytosis. In here we got some receptor-mediated endocytosis, in which case a receptor is like, I want that protein. Let's grab on. There's a lot of things going on here. I don't know how important everything is. But those receptors are connected to adaptor proteins, which facilitate the accumulation of clathrins, clathrin codimers, which form your clathrin-coated vesicle, which eventually goes over here. And also, how important is this? I don't know. but in the lipid bilayer, there are some lipids that are special for adaptive protein binding, and those are called PIP2 lipids. Moving on. Okay, so let's say I get a thing and I decide that I don't want it anymore because it's been ubiquitinated, someone decided that it's bad, and if it's ubiquitinated, it's time to go. So I will go to a multivesicular body, also known as the late endosome, which comes from the transcriptome network or early endosome. So now I'm this thing. In this example, there are 4 proteins. More likely, there are a lot more than 4 proteins, but yeah. The ones that have been ubiquitinated, they gotta go. The ones that haven't, we want to keep. So eventually, we want it so that these stay in this exterior membrane, so that when we fuse with the lytic vacuole, it just becomes part of the tonoplast. And these that have been ubiquitinated are on the inside, so they are inside the lytic vacuole and they can be degraded. So what we have is escert1, this enzyme that aggregates all the ubiquitinated proteins, puts them in one place. And then we'll have the invagination of the membrane, which is done when escert2 recruits escert3. Escert3 will in fact have this membrane deformation. You'll have what's called an intraluminal vesicle, or ILV. And escert3 will also deubiquitinate them because we don't want to put our ubiquitins in the lytic vacuole. Those are valuable, we can use those. But now, everything in ILV, you will usually, I've never seen a multivesicular body drawn with one ILV. You usually have several. In plants, they're connected. But for the simplicity of this diagram, we have one, and we want her to be degraded. And I did not redraw her over here, but you get the idea. So if we want to fuse this multivesicular body with the lytic vacuole, you need to have some way to grab. This goes for all the things when we have like one membrane-bound thing associated with something else. This is just the time that we talk about it. It's going to have a thing that's going to grab on and say, come here, I want to fuse membranes with you. Aren't we in love? That's called the Rab effector. It'll bind to Rab GTPase, and then GTP hydrolysis, and it'll kind of like pull it in, and then these snares will help the membranes fuse. There's the V snare, vesicle, vesicle, right? V is for V, vesicle, and then T for target. Yeah, the vesicle going to the target. Yeah. Yeah, that's what I have to say about that. Tonoplast is the membrane of the lytic vacuole, which is very acidic because we'd be lysing things. In animals, this would be called the lysosome. And proton pumps would be maintaining that. Also, they have specific.
Very good.
So the tonoplast, remember the lytic vacuole membrane, will have aquaporins that start with T for tonoplast, and the plasma membrane will have ones that start with P for plasma membrane. So if you're looking at it in fluorescence microscopy, never wrote anything else there, you can see the difference between just like a circle shape if it's a lytic vacuole or the plasma membrane, because the plasma membrane is going to be the shape of the cell, but the vacuole is going to be the shape of the cell minus the stuff that is like pushing away, like the cytoplasmic components, including the nucleus. So this one, the vacuole, will have like this different shape. At the end, we'll talk about who's that Pokemon and other, like being able to recognize through microscopy what a thing is. But a lytic vacuole isn't the only type of vacuole. You can also have a protein storage vacuole, which obviously you do not want a pH of like 2 if you're storing proteins, because that's not a good protein. So that's going to have a neutral pH. And how do you make a vacuole? You have 2 ways. One is from ER, which is a pattern we will see repeated, just like budding, or you can have them from MVPs. Fun facts. Speaking of things coming from the ER, you can have lipid droplets coming from the ER. So lipids will accumulate, seep in, will decide if it's going to bud off, regulated somehow, I don't know how. And there's a monolayer here. Notice how there's 2 layers here and 1 layer here because we're full of lipids. We don't want to have a phospholipid like pointing back inward to contents that are hydrophobic. That's silly. So it's just 1 layer of, like the phospholipid head would be on the outside and the fatty acid tails would be on the inside, filled with lipids, including triacylglycerol is the most common one, or TAG. That's lipid droplets. Moving on. Peroxisomes, I love peroxisomes. They'd be breaking down fatty acids. They'd be managing photorespiration by turning phosphoglycolate into glycine. They'd be dealing with ROS, maybe converting more dangerous ROS into hydrogen peroxide, or just having hydrogen peroxide turned into water. And you can, more microscopy, recognize a peroxidome specifically because it has this urate oxidase crystalline core in the middle. It's also perfectly round, fun fact, unlike your vacuole. So in electron microscopy, this will appear black because it's very, very concentrated. So if you want to review, who is that Pokemon? These are the things. So who is this? Who is that?
Who's that Pokemon?
It's the plasma membrane!
Ew.
Okay, don't look at it. Who is that Pokemon?
It's our Totoplast!
Cool. And then who did we just talked about her?
Who's that peroxidome?
Peroxisomes.
Okay. Yes. Okay. Now let's talk about protein import. This might seem scary, but like, just draw it out, and I feel like you're gonna be okay. So if I'm to be imported, if I'm a protein and I belong in the peroxidome, I'm gonna have some sort of sequence indicating that I belong in the peroxidome. That's gonna be your signal peptide. In this case, PTS, peroxidome transit sequence. I don't know. There's one on the C-terminus and two on the N-terminus. Not two different ones. You will have one. It will either be on the C-terminus, PTS1, or you will have one on the N-terminus, PTS2. This one is associated with PEX5. What does that mean? PEX5 is kind of like important beta, but instead of for the nucleus, it's for the peroxidome. So this is I never wrote it, but it's pretty obvious. This is the peroxidome. If I detect PEX5, PEX5 is like, you know, the way that the NPC recognizes an NLS, or it recognizes important beta. PEX5 will be like, I am allowed to go through this channel and go into the peroxidome. This channel is called PEX13. PEX13 is going to be like, come on in, baby girl, I know you're good at PEX5. And then this protein that has this signal peptide is good to be free, go wherever it needs to be in the peroxisome. It's very reliable, also not cleaved. Same goes for the NLS. And it'll be free. But PEX5 doesn't want to live in the peroxidome. It's got to go searching out for more peroxidome destined proteins. So she'll be recycled. So PEX14 is going to grab her and send her back through another transmembrane protein to go out. That is PEX21012. And on the way, she'll get ubiquitinated. This ubiquitination is important because PEX1, PEX6 is an ATPase that will only work for ubiquitinated proteins. So it will then pull out our ubiquitinated PEX5 and release it back to find more things as recycled. But if it's searching for PTS2, it'll be PEX7. Let's go do a quick compare, contrast, nucleus, peroxisome, protein import. NPC is the complex that things go through to come in. That's about 1,000 proteins. I don't know why I said thousands. About 1,000. Whereas in the peroxisome thing, the channel coming in, it's just PEX13, so one protein. The single peptide is the nuclear localization signal versus PTS1 or 2. The thing that recognizes it in a nuclear pore complex are those FG loops, remember, and they bind to heat motifs. Whereas the peroxisome, PEX13, has YG repeats that recognize the WXXXF-/Y motif. Do you have to memorize that? I don't know, but it's there. And they have a friend that they recognize. Important beta is what is recognized by the FG loops letting through the nuclear pore complex into the nucleus, whereas the peroxisome will have PEX5/7 as their friend. Say, yeah, on it. You're all good. And then how do we get to a peroxisome? We can have either budding from the ER, again. Usually there will be like the integration of a protein that says like it's time to bud off and turn into a peroxisome. Then we'll get a pre-peroxisome, and then that'll turn into a mature peroxisome. Or option 2 is we can have a peroxisome like doing fission, like kind of like a bacterium does. So first it'll get really long, and that happens to PEX13 or PEX11, another PEX. PEX11 will induce tubulation, and then you want to have your individual peroxisomes. That'll happen through, so budding and fission will happen through Phys1AB will recruit They're membrane anchors. They will recruit diamond-resembling proteins. We didn't really talk about this, but diamond is something that helps budding in clathroid vesicles. That's something from the first part of this class. So diamond-resembling proteins will help the budding of our peroxisomes, and then we have more peroxisomes. That's it. Do you still remember? Is that Pokemon?
Beautiful. Oh, Pokemon. Look at that.
I don't know. I'm not doing it again, but.
No, yeah.